Can you stop sharing for a second? Otherwise, we won't be able to.
Okay. Where is the?
Let's see.
Yeah.
Okay. First Sunday of the month, 11 o'clock, time for chai and wine. Thank you. It's a rainy day. Thank you all for coming here. And thank you, online audience, for joining us on Zoom or on YouTube, wherever you're watching. For the benefit of the online audience, please put in your questions in the chat on YouTube and the Q&A box if you're watching on Zoom. So today we have a very interesting topic by Sneha Hegde, who is a research scholar, a PhD student senior PhD student at TIFR in the Department of Biological Sciences, where she works on some tiny creatures that she'll talk to you about. Sneha, you should also give them an introduction about your path into TIFR before you start. And the way we'll do this is we will have she will tell us something on screen, I guess. And do feel free to ask if you have an important question. Just please raise your hand and we'll take the question. Please repeat the question if they ask, because otherwise the online audience won't know what they've asked. Please repeat that. And yeah, after that, we will take a break, have chai, and we will then stop the live stream. Sorry, guys, you don't get chai if you're watching it online. And then we will come back here and we can have questions, more questions after that. Okay, and I'd like to thank Shweta, who's here and is also going to help answer questions on Zoom and YouTube if there are. She'll help us answer them, some basic stuff, or she'll tell us that there are interesting questions coming up. And yeah, all yours. Powerhouse. Let's going. I'm monitoring YouTube.
Okay. So I hope everyone can hear me at the back. Hello, everyone, and good morning. I am Sneha Hegde, and thank you for attending this session, coming here, and all of our online viewers for attending from their homes. I'm a PhD student, as Arnav said, in the Department of Biological Sciences, TIFR Mumbai. And before coming to TIFR, I had done my bachelor's in biotechnology from SIS College in Mumbai. And then I went to do my master's again in biotechnology from MSU Baroda. And then I landed up in TIFR to do my PhD in neurobiology, to be more precise, cellular neurobiology. That is, we see what happens inside the cell using neurons as a system. And then we discover beautiful stuff. Some glimpse of which I'll be showing you guys, everyone over here now in today's session. So in today's Chai and Why, we would be talking a bit about mitochondria and discussing about all the things that mitochondria does in the cell, particularly in neurons. Before I start this session, I'd like to ask a question to everyone who's come here. How do you think we get energy to do our daily work? For example, things like playing, exercising, or working for students going to school. And spoiler alert, it's not mitochondria. The answer is not mitochondria, and there is no scientific answer to this question. Oh, very good. Okay. Sorry. Yeah, what were you saying? Go on. Okay, we got an answer. It's whatever we eat, which is the food. And yeah, the food that we eat. So yeah, it's Jerry. Okay, so if everyone might have noticed, whenever we feel hungry, we feel a bit tired. And then we eat something which can be as small as a Snickers bar. And then we get a slight amount of energy to do our next task in hand, right? So has anyone wondered what happens when we eat? We get energy, yes. Very good. But how do we get that energy? Okay, we have a very smart crowd over here. Let's see the upcoming slide.
Repeat some of the answers.
Huh?
Repeat some of the answers.
Okay, yeah, sorry. I'm sorry. I'll just. So the answer that we are getting here is quite correct and varied, wherein a very smart kid answered that it's the energy that we get out of food that helps us to do the next task. And a young gentleman suggested that it is a glucose that we get from the food that we eat that helps us to sustain or in turn get the energy to do the next task. So let's see what exactly it is. Is it about the food that we eat that gives us this energy and also the glucose? So the food that we eat on a daily basis, which can range from the breakfast in the morning to a chocolate in any time during the day, or any junk that we eat during the day, enters into our body through a mouth, through a place called mouth, which is an opening in the body. And through the mouth, it enters into the digestive tract, which is shown here. Now, this food is actually a mixture of various complex substances put together in one particular unit. As it passes down to our digestive tract, which is shown here in this image, it is acted upon by various chemicals, various enzymes, to break it down into a very small, into its smaller constituents. Now, our body likes to keep it simple because, why complicate things when you can get done with simpler things? So as this food gets passed down through the digestive tract and it's broken down into its different simple subunits. And one of these subunits, as the gentleman over here rightly suggested, is glucose. And another components of this food comprises of amino acids, glycerol, and fatty acids. But then all of this thing happens in our digestive tract, right? And our entire body needs energy. So what happens after these things are digested into its simpler components? So everyone over here must be aware of this delivery system like Amazon and Flipkart, wherein you order something and things get delivered onto our doorsteps, right? Our body also has some kind of delivery system like that, wherein the nutrients which are broken down into smaller components are absorbed at the smaller intestines, and then through the blood vessels, which act as the transport tract for them, is passed on to the basic unit of our body, which is the cell. Okay. Now, what happens when these subunits enter inside the cell? Now, there are several different things inside the cell, which includes bigger organelles, smaller organelles, DNA, nucleus, and the broken down nutrients which enter in these cells, which are delivered through the digestive tract, are then distributed within these smaller subunits or smaller organelles, which then utilize them to perform or to enable the cell to perform its daily activities. We are not concerned about those today. What we are concerned about that is what mitochondria does, because we'll be looking a bit more about mighty mitochondria in this session. So the kid was right, we get energy, and sir was also right, we get glucose. But how does mitochondria produces the glucose that is produced, that is delivered into the cell and produces energy? Let's just get a small glimpse of it and then move on. Now, please stop me in between if there are any questions. If you guys don't understand anything, I'm happy to answer the questions. Okay. Before we dive deep into the functions of mitochondria, in the session today, we will not only be looking at mitochondria as the powerhouse of the cell, but we'll be giving a different perspective of mitochondria does in the cell and the impact that mitochondria has because of its functions on the cell using neuron as a system. Okay, but before we dive deep into this subject, let's see what mitochondria is made up of. How does it look? Now, this is an image of mitochondria when we cut it from the center and cut it open. But in reality, mitochondria is a cell inside a cell. Why do I say so? Because it has two layers. It is protected by two layers, which is the outer membrane and an inner membrane, wherein the outer membrane protects the mitochondria or separates it from the rest of the environment within the cell, and the inner layer protects it from the liquid that is present inside the mitochondria, which is famously known as the matrix of mitochondria. But there is more to it. This is one of those organelles which also has its own blueprint, that is the DNA. Okay, so the next question that I would like to ask here is that where do you think we get mitochondria gets the DNA from? From which parent? Mother, father, or from both the parents, like our DNA is in the cell? Both the parents. Okay. Can we have more answers? Sorry? Mother. Okay. Yes. Mother. Anyone more? Anyone else? Do we have any online answers?
Let's see. Nothing here.
Okay, great. We have a very interactive crowd. I'm ready.
I'll just I'll put the question.
Yeah. Okay, so let's move ahead. I'm sorry, but the answer.
Yeah, there are some answers on.
Okay, great. We have a very smart audience today. Nice. So the DNA that is present in mitochondria is obtained from our mother. It's not just the DNA, but all the constituents of mitochondria, including the mitochondria itself, is obtained from our mother. The only thing that we get from a paternal side are one set of chromosomes in the cell. That's it. And apart from that, mitochondria also has its own production unit, that is the protein synthesis unit, which uses this mitochondrial DNA as the blueprint to produce its own proteins. Interesting, right? That's why it's a cell within the cell. Now, how does mitochondria produce energy? We came to know a lot about what mitochondria looks like, what are, we glimpsed on the structures of mitochondria. But how does a small organism is capable of producing energy to energize our own entire body? We'll just skim through it. There are a lot of steps involved in this. I'll just skim through superficially what it does. Oh, sorry. Yeah, I'll skim through superficially what it does, and we'll come back to it. Okay, so what mitochondria does is that there are a set of proteins that are present on its intermembrane chain. As you can see here, these 4 proteins, which are quite efficient and placed very smartly one after the other, so that the glucose that is produced, that is delivered to the cell, enters inside the cell, is further processed, then further enters inside mitochondria. Then mitochondria extracts the charges to build up a charge around this membrane. And there are a lot of other components which are passed from one protein to another, which can be seen over here. In turn, finally,
so which is the outside of the cell, down or up?
Yeah, sorry, the outside of the cell is up. Not the outside of the cell, the outside of the membrane. So this is the membrane. Okay, I'll point it over here.
Go back and maybe start this video.
Yeah. So this is the inner membrane. This is the space between the inner membrane and the outer membrane. And this is the matrix of mitochondria. Okay. So what happens is that all of the things that are extracted then are passed by extracted from the nutrients, which finally enter into the mitochondria, are passed into these proteins, and these proteins then carry out, like, do a systematic this process called electron transport system to transport charges from one protein to another, and produces the final product called ATP. This ATP is actually an energy molecule, which is then dissipated in the cell and fuels the cell. Now, if one cell gets such energy, imagine by one mitochondria, imagine there are a number of mitochondria present in a single cell to fuel that cell. And then even each cell is fuel in our body, we get energy. So that is how we get energy. Yes.
So there's a question from the audience. Abhinav is asking, can we increase the mitochondrial electron transport in any way and make it even more efficient?
We cannot increase the mitochondrial electron transport, but we can eat good food to get more energy and make ourselves efficient. Yeah.
So up until this energy is converted into ATP, it's not usable by the cell or mitochondria up until this point, right? So in the ATP that both the mitochondria and parent cells can use, like their energy now.
Yeah. Yeah.
Yeah. Okay.
I don't think I got your question.
So the energy that was like supplied through blood vessels, all the way to the cell, is sent to the mitochondria, and then it gets converted into ATP. And you said that ATP is the usable form of energy.
Exactly. Yes.
So that goes out for the cell also.
That is the one that goes out for the usage of it. Yeah. So the question was, whatever we eat right now, that is that a usable form of energy, or how, or whether the ATP that is the final product, is the usable form of energy? and whether that acts as the source of energy for the entire cell, and the answer is yes, that is the case. Okay, so yes.
Is this only secret for protein or including fats and other substances also?
Yes, so there is a very complicated pathway through which all of the macromolecules that are ultimately broken down in our body are used for energy production. But there is a level at which each stage at which each and every macromolecule would be used. So, for example, glucose would be used first. And if we don't have any of the glucose levels that are present, then the body turns out to the next macromolecule, which is easily broken down. And then the body turns out to the next macromolecule. So it is like that. And that is why we can survive for a longer period of time when we are fasting. So if we don't eat anything, this along with the things that we eat are stored in our body. And then that acts as like a storage, a backup nutrient content to which our body moves to, which are then moved out and then changed into its different format and transported into the mitochondria so that it can use to produce energy. And there are also different ways in which we can have quick sources of energy also.
So what mitochondria is taking from the cell is glucose.
Glucose is actually converted in a way. So there's this thing called TCA, Krebs cycle and all. So which is again very complicated and has a lot of steps. Glucose is converted and then enters into mitochondria. So because mitochondria has these barriers, right? Outer membrane and inner membrane. So it needs whatever is entering in the mitochondria should be permeable. It should be in a way that it can pass through the barrier of mitochondria. So glucose is converted. There is an intermediate step in between through which it enters the organism. Okay, great. So here, after this, I wanted to okay, let's get back to the presentation. Yes.
So the request from the online audience is to please repeat the question.
I'm sorry.
So I think the question was, what does glucose? Can
Okay,
glucose
yeah.
enter the mitochondria?
When glucose enter the mitochondria.
Yeah, just
so there were two questions. I'll repeat those two questions. The first question is whether glucose is the only macromolecule that can be used as a source of energy for us, and the answer is that it is. There are different forms. All of the macromolecules are broken down at different levels, and can be used, and then are used for us to produce energy. But it all depends on whether the availability of glucose is there or not there. But glucose is the primary macromolecule that is used for the production of energy. And the second question was whether glucose directly enters mitochondria or no. And the answer is that there is an intermediate step because there are two barriers, that is the outer and inner membrane. Glucose needs to be converted into a form which is capable of entering inside the mitochondria, and then it is used by this organ. Okay, great. I'm so sorry. I keep forgetting to repeat the questions. But yeah. So is mitochondria just restricted to the source of being the powerhouse of the cell, or does it have some other functions? To answer this question, scientists use a lot of different techniques, but the technique that I'll be focusing on today and that we do very heavily in our lab back in TIFR is to mark the object in the process that we are interested in by various dyes, which we very famously call as fluorescent proteins, and then look at them or observe these objects and processes under different conditions using a microscope. So, how does mitochondria look like under a microscope? These are mitochondria in the different projections of a neuron, which is like a cell that is present in the brain. And these punctae that you see here, are mitochondria within the axon, which is one of the main projections of a neuron. We'll come back to what a neuron is in the next slide. And this is how mitochondria looks like in the skin cells. So if you see, there is a difference in the way mitochondria looks like in one cell as opposed to others, wherein in the projections of one cell, that is in the neuron, mitochondria looks in a very punctate manner. But in the skin cells or in normal cells, mitochondria looks like thread-like structures or tunnels present, which are dispersed throughout the cell, right? So this also shows us that mitochondria has different forms and shapes, And there is a research that is going on in the field to understand why does mitochondria has one kind of form in one cell or one part of the cell as opposed to the other cell, and whether it is associated with a different functionality of mitochondria. Yep.
So in that image you showed, did you do something to make those mitochondria glow? Because the second image, they're not glowing. The first image, they're glowing.
Yeah, so in both of the cases, so the question is in all both the images, in the first image, it appears that mitochondria is glowing. And in the second image, it appears that mitochondria are not glowing. So is there something that we do that we did in order to make the mitochondria appear to be glowing? The answer is that here we've not done anything. We just use fluorescent proteins to visualize the mitochondria that are present. Fluorescent proteins are basically proteins which are lighted up when you shine them or when you expose them to one particular kind of light. So when, say, the light that we see around us, it has a lot of wavelengths associated with it. When you take one component of that light and shine it to a fluorescent protein, it starts lighting up. And that is what you are seeing over here. Okay. So do mitochondria actually are only the powerhouse of the cell? This is the question that I've been building up since the past few slides. And the answer is right now here. I just want to get a sense of the audience here. So is everyone aware of calcium that we find in our bones? Okay. But calcium is one of the most important components of our cells, which apart from bone, which makes our bones stronger, is present in the form of ions or in another forms with other chemicals, which act as a very important signaling component in the cell. So it relays a lot of information and enables the proteins downstream present in the cell to work. Okay. But when there is an increase in calcium, anything more of any particular thing is not good, right? So more of calcium is also not good for the cell. And whenever there is an increase in calcium, the mighty mitochondria comes into rescue for us. So I would like everyone, I'll be playing a video right now. I'd like everyone to look at the video very carefully. What I'll be playing is that, so there is a thin line over here, which you can see faintly, which is a projection of a neuron that is in the axon, and the puncta are mitochondria, as I had shown everyone earlier. When I play the video, there will be a flash of a signal that would come and go. That flash is an increase and a decrease in the calcium. And I'd like everyone to observe the mitochondria both before and after this flash. Okay. See? Should I play it once more?
Yes.
Okay, it's on loop. Okay. So what happens to mitochondria before the flash and after the flash? Increase in size. To red to yellowish. Okay. Basically, there's a change in intensity or the way it appears. Became whole axon became black. Okay. Okay. Okay, great. Nice. Good observation. Okay, so I'll go on. I'll repeat the answers that the audience said. One person said that the mitochondria increase in size. One person said that the color of mitochondria changes. One person said that the neuron becomes a bit darker. The neuron was slightly visible before, but right now it's become a bit dark in color. So what happens here is that the mitochondrial outer membrane has pores. So just like, we have doors for our houses. If we want someone to enter inside the house, we open the door. If we don't want that person, we just close the door and pretend that we are not in the house, right? So in a similar manner, mitochondria has doors for several such molecules present in the cell. Now, whenever there is an increase in the calcium within the cell, this door opens for the protein and the excess of calcium comes inside the mitochondria. So it acts like a sink for the mitochondria, helping to maintain the level, sink for the cell in terms of calcium, helping to level the maintain of calcium within the cell. As a result of which, the marker that I used here is actually a calcium marker, which marks both mitochondria as well as calcium. And you see, whenever there is an increase in calcium, the size of mitochondria appears to increase. It's not actually the size of mitochondria increasing, but now more calcium is present, enters inside this organelle, as a result of which you see it to be increasing and the intensity to be changing, and the neurons calcium level goes a bit down. So in a way, it helps the cell to protect from excess of calcium. But the function of mitochondria is just not restricted to that. Everyone must have fallen down in their life one or the other time, right? Cut, injury, skin injury, something like that, right? Mitochondria also helps in the process of healing this wound. How? Let's see. So whenever you have a wound in your hand, a cut in the hand, there's a slit, and that slit starts closing on its own without you doing anything. What we actually do is just add a turmeric as an antimicrobial so that the infection is not increased or severity is not increased, right? But the actual wound healing process is done by the body, right? Mitochondria is one of the factors that enable the skin cells to heal this wound by producing superoxide radicals. Now, superoxide radicals are considered to be the villains of the cell, wherein they are known to kill the cell. But if they're produced at a very controlled level, they can help the cell to function properly, and that does happen, and mitochondria does. So I'll show you a video again once, and let's see what happens and what mitochondria does when the cell is wounded. So the video here is the video of the epidermal cells, that is the skin cells of C. elegans. And now when I'm playing the video, this is the site of injury, wherein you put the laser and there is injury. And when you look at the mitochondria here, they're flashing. Can you see the flashes? Can you see the flashes over here of the online audience in these places? So the marker we've used here is a mitochondrial specific radical marker, superoxide marker. So only the superoxides that are produced by mitochondria would be visualized by us. What happens is that whenever there is wounding or whenever there is injury to cells, mitochondria around the region where the injury has happened starts producing these radicals, but at a controlled level. And these radicals then send a signal downstream in the cell that, okay, there's something wrong with the cell. You need to initiate the process of healing. And researchers in the field have seen that if you stop mitochondria from producing these radicals, the process of healing of this wound is also delayed. So yeah, mitochondria plays this role as well. Yeah. Yeah. Yes. Yes. Yeah, so the question was, is that a single skin cell because of the density of mitochondria in this cell? And the answer is yes. Over here it is, but people can see multiple cells under the scope and then see a similar kind of signature that, they're just present everywhere. Yeah.
For the external injuries that you're talking about now, it was a deliberate wound.
Yes.
To a laser, right?
Yes.
So is it all surrounding cells of any type, or is it like some like white blood cells type, the ones that are actively sent to the injury point to injure?
So no. So the question is that whether this effect happens to all the cells around, or whether this production of superoxide radicals after injury happens only to platelets and white blood cells. The answer to that is that people in the field have only seen it happening in the skin cells. So yeah, so in the skin cells, the mitochondria which are present in them, in the epidermal cells, produce these radicals. And then these radicals act as signaling molecules, which then pass on the message and activate the other proteins to heal the wound. There are, again, so that's why when I said that it is one of the factors. There are a lot of factors which will then send the signals to these neutrophils, platelets, and all of our immune cells to come to that place and then, obstruct the infection and so on and so forth. But that is a different thing. And mitochondria plays a role in that also. But we can talk about it more after during the chat.
Can see that there's a lot of difference between the image 1 and the image 2. How do you identify that both are mitochondria?
Okay, so the question is that there is a lot of difference between image 1 and image 2 in terms of mitochondria. So how do you identify that both are mitochondria? The simplest answer that I would give right now is that we use markers that are specific to mitochondria. So we have techniques through which the proteins that we make, the fluorescent proteins that we make, we tag it with a signal that will deliver it to only mitochondria genetically. Also, mitochondria has its properties like the charge that it has, and it's like, every person, even though we all are humans, we have certain characters, and using that character or the facial features or the physical appearance, we determine whether this person is different from the other friend of mine, right? In a similar manner, mitochondria also has its own characters, and we make use of those characters like a different charge, has a certain kind of membrane, and through that we use dyes which will selectively go and bind. But currently in the field, with the advances that we have in science, we make use of fluorescent proteins, which has to which we attach a mitochondria specific tag, which only labels the mitochondria and nothing else. Okay, but one thing that I'd like to highlight here, and thanks to you, sir, is that mitochondria in the projections of a neuron look punctate, but you will see these kind of mitochondria in the cell body. What is a cell body? I'll come in the next slide. But these kind of mitochondria are found in the cell body. And in the projections of the neuron, you will see punctate mitochondria. Okay. Okay, so the question that is being asked, that is asked now, is that the thread-like mitochondria that we look at over here, is that a single mitochondria or are they different mitochondria? Answer is they are different mitochondria, but a single mitochondria. So I'll point out over. So if you see this can be a single mitochondria. But then there's a very small space between the ending of one mitochondrion and the beginning of another mitochondrion. So looking at it, I say, on a resolution like this is quite difficult. You need really high-end confocal microscope to actually look at whether they are more than one or one. But this definitely is not a single mitochondria. Yes. Oh, I saw.
Are they the same throughout the body or?
No. So the question is how many mitochondria are present in a single cell, and is the number same for all the cells in the body or do they change? The answer is quite difficult, sir, because it depends on what kind of cell we have. So if it is a very energy demanding cell, the number of mitochondria increases. So the cell itself amps up the production or the mitochondrial biogenesis, as we say, which is production of new mitochondria, which is done by cutting off the existing mitochondria and adding new proteins in the mitochondria itself to produce more mitochondria. So it itself undergoes a process of breaking itself down and producing more of it to provide more energy to the cell. And there are many in a cell. So it all depends on what kind of cell you're looking at. So if there are skin cells and like in this situation wherein, there's injury and there is requirement of more energy to help in healing of the wound or something like that, the number of mitochondria increases. But then it would go down if the cell thinks that, okay, now we don't need so much of energy production. Okay, great. And because of these functions, mitochondria not only is a powerhouse of the cell, but it plays a very central role in the signal processing within the cell itself. Now, these functions are useful not only in smaller cells, which are compact, spherical, or rectangular in shape, but they are very much useful in cells like this, which is the neuron. Now, if you, as you can see in the slide here, instead of being spherical in shape, neuron is like this massive human-like structure, very complex architecture, containing its head, which is also known as the cell body over here, and various projections moving out of the cell body downstream and like in one direction over here and in other directions as well. Now, the biggest projection that you see is the axon, which is like a projection that helps this particular cell to communicate with its neighboring cell. And the smaller projections that you see, which are jutting out from the cell body, is known as a dendrite, which receives the information from its neighboring cell. So it is like a cell which is capable of talking to its other cells, and it has different compartments. It's like, a telephone exchange company. or some department which receives signal from one end and emits out signal from the other. Now, these neurons in populations of millions of cells in millions of numbers are packed together in this wonderful organ called the brain, which is very complicated. But if you take a small slice of the brain, color it using a mitochondria specific dye and visualize it under the microscope, this is what you see. Amazing, right? And each neuron has the capability of forming connections with its neighboring neurons and then talking with one another to actually relay the information like, okay, now your hand has touched something hot, you should pull it off, and rather than keeping your hand on that hot, something like that. And mitochondria, with the functions that we have seen previously, are quite important, are found to be quite important for the normal functioning or the normal ongoings of such cells called neurons. And there is a very active research in the field to understand what is the importance of mitochondria in the neurons. Now, we in lab try to understand this question by using these very simple and tiny or model organisms called C. elegans. Now, what are C. elegans? C. elegans are worms which are usually found in the soil and they feed on bacteria. Now, the best part of these worms are the biggest size of this worm is 1 millimeter, like the adult one. Now, as you can see in this slide, they're transparent also. So when you put them under the microscope, you can actually see through its body. There's no privacy for this one. So there are intestines. If you see these things, these are the intestines of the worm. This is the mouth of the worm. This is the tail of the worm. This is the anus of the worm. So you can see everything because it is transparent. And as a result of which, when you want to study certain processes, you can just label whatever processes that you want to see, and then put it under microscope. And by fluorescent tagging, fluorescent labeling, like the one that we looked at mitochondria, you can live visualize these different processes without even cutting open the animal. So that is a very good advantage. When we see these animals under the microscope, this is what you see. So it is quite easy to maintain these animals without using a lot of space. And this is how a glowing worm looks like. So when you add a fluorescent marker in the animal and look at it under the microscope, you will see some glowing worms like this. So these models are quite useful to understand the processes that happen within the cell, and then extrapolated to human context as well. And the other best part of it that all of the neural connectivity, that which neuron connects to which within the worm, is already known. So any process that we change or any process that we perturb, what is the implication of that process to the neuron becomes easy, because there is a lot of information that we already know about this system previously in the past. Okay, so now what we do is that, as I had said earlier,
I mean, you said the brain is very complex, and there are billions and billions of things. How complex is this guy?
it just has 302 neurons, and the brain of the animal is over here. In this case, I mean, it's not a brain, but it has a meshwork of a lot of neurons present in this head region, which we call as a pseudobrain of the animal. And because it has 302 neurons, it becomes quite easier to study what happens within a neuron. And because the basic functioning of the neuron is conserved in all the phyla, we can extrapolate it to human context. Okay, so now let's see how a mitochondria actually looks like in a neuron. What we do is we use a fluorescent protein, tag it with mitochondria. So only the mitochondria in the cell are glown. And this is what we see. So the cell body that I had shown everyone earlier is over here. And here is where you'll see the tunnel-like mitochondria. And if you see, the axon contains only punctate mitochondria. Now, the other obvious question is, so does this remind everyone like a road or a rail track in which there are cargoes or packages in between these road or rail tracks? So the next thing that people would want to see is that whether they are just sitting there like houses along the tracks, or whether they're moving like vehicles along the tracks. So what we do is we take a part of this neuron, we image a part of this neuron, take video of a part of this neuron, and see how mitochondria look like under this image. And we see that 8 out of 10 mitochondria are stationary, they don't move at all, and only 2 out of 10 mitochondria are moving. Okay, so it's quite interesting, right? That given the entire thread of the neuron, you see mitochondria being present throughout this entire thread, which is the axon, but there are only few which move, and the rest of them are stationary. So there is another field that another question that is being pursued in the field, which is to understand what is the importance of having mitochondria along these neurons stationary. And we'll come to that later. So up till now, we were talking about the function of mitochondria. Now let's see what the impact that this has in the cell. And the cells that I'd be specifically talking about is the neuron, because of its huge complex architecture, which helps us to actually understand what impact does our organism has on such cells. To understand that, we use two systems. Easy one, which has mitochondria in the neurons all over the neuron, and the other system, which does not have mitochondria in the neuron. So it's yes or no. If there is an effect on a function, we'll see that clearly in a system wherein there is no mitochondria. And if there is an effect, we will, basically we'll see the effect in the system where there is no mitochondria. Okay. So our system, we were particularly interested to study this entire phenomena called as growth after injury, which is technically known as regeneration of the neuron. To get into the details of it, whenever we undergo an accident, like a car accident or anything of that sort, or whenever we fall, our neurons break, because you saw that these are long thread-like structures. And when these neurons break, certain neurons of our body, like the neurons that are present in our peripheral nervous system, which connect our hand to the brain, are capable of growing. But the neurons that are present in the central nervous system, which are present in the spine and in the brain, do not have the ability to heal itself or grow after it is cut or injured. So there is an active research in the field to understand what are the factors that help a neuron to grow, so that we can use that as a therapeutic treatment for regenerating or growing back the neurons of our spine and the brain. In turn, curing things like paralysis, which people often face when they undergo any car accident or any injury. Okay, so whenever a neuron is injured, it's cut into half, and then it undergoes growth and is capable of forming connections with its neighboring cell or the neighboring neuron. And when the neuron cannot grow, as I just explained earlier, the connectivity is lost, as a result of which it cannot communicate with its downstream neuron or the cell. And as a result of which, one of the adverse effects of this end in the communication is paralysis. And one example that some of you may know is this famous actor who played the role of Superman, Christopher Reeve, who faced paralysis just in his entire body below the neck after he fell down off a horse and broke his neck. That was because his neurons in the spinal cord were all injured, and because these neurons cannot grow back, he underwent a full body paralysis. Okay. So what happens in the neurons which actually grow? When the neurons are cut into two different halves, there is this structure called growth cone formation in the region where where the neuron gets cut. Now, this growth cone is a very complicated structure, but imagine it as a hand of a monster, which is now trying to move in an area to see which area it can explore where there are people and which area it can explore where there are no people. Okay. So what this growth, it looks like that actually. It looks like a hand which is actually moving through a surrounding. And then when it actually finds a contact with its neighboring cell or another immediate neighboring part of the neuron, it will start forming contact with that neuron. And what people are trying to do is to understand how is this growth cone being formed, or what helps this growth cone to move on. Okay. And once it understands that, okay, I can form a proper connection with its neighboring cell, it regrows into its normal cell, which was earlier, regaining the functional connectivity.
Is going blind to touch the muscle cell or something. So if it touches the wrong muscle cell, then the virus will be wrong.
Yes. So the question is that how does it know that it is forming the right connection If Galti say it goes and knocks the door of a wrong neighbor, it will be a guest in the wrong neighbor. That's right? Yeah, it does not know, actually. But in certain cases, so this is a very highly evolving and exploring field. In certain cases, the cells very well know where exactly to go when the neurons cut. And the people, how should I say? Okay. So the answers to this question is that in certain cases, the cell does not. But in certain cases, there is a gradient of signals that are released by the other cell, and that acts as a guidance cue for the cell which is injured to move in that direction. So it's a very gray answer to that. If that guidance cue is absent, it won't move on. I mean, it will move in a lot of directions.
I have a question. One of the slides before this,
Okay.
you showed two neuron cells. So one of them had the mitochondria in there, and look, the one, yeah, this one. So one had and one did not have. The one with the did not have, you have removed it manually. The mitochondria.
No. So we use. Sorry.
Repeat the question.
Yes, sorry. So the question was that in this particular slide, the neuron which does not have mitochondria, how do you achieve this? So these neurons are quite small. They are in the scale of micrometers, which you, as a result of which you cannot do anything manually with them. What we do, what we have done in this case is used a protein which helps in the movement of mitochondria in the neuron. And we have done some changes in the protein such that now the protein cannot function. Because of which now we only see mitochondrial signal here in the cell body, and the entire neuron doesn't have the mitochondria. Okay, you had a question. So the question is that these mitochondria over here are there, but they are not functioning. The thing is, we don't know because we do see mitochondria in the cell body, but because they are not dispersed within the cell, we can't really say, we need to test whether the mitochondria which are in the cell body, isolate them and see the functionality of the cell, which we haven't done yet. Okay, so the next question is that when you cut the neuron, the lower part, which is also called as the distal end, a distal part of the neuron, does it undergo degeneration or does it, sorry, help the other side of the cell, that is the proximal neuron, to form the growth cones? The answer to that really depends on how fast the growth cone is formed. So up till now, people have not seen the role of this distal part on the growth cone formation, or at least I am not aware of that research being happened. But if it takes a while for the growth cone region to form and move ahead, it undergoes disintegration. So there is a process of disintegration of this distal part initiated as soon as you cut the neuron. But if the growth cone moves faster, like it's formed faster and it starts elongating in a faster rate, then it is capable of rejoining to its distal cone. We'll come to that slide now. Okay. We did this. Okay, so we were interested to see whether mitochondria play a role in the process of regeneration. And not only we, a lot of labs all over the world are actively trying to see how mitochondria help in this process of regeneration. So what we do is, we have a worm, we can easily see the neuron inside the worm. And we have the tools to cut a neuron using a laser. So we cut the neuron with the laser and we simply observe for regeneration. Now, if there is any regeneration, this is what you'll see. So this is a neuron which was cut and it was capable of growing. So this is the growth cone-like structure that I was talking about will be formed. And this is the distal tip, distal end of the neuron. But if there is no regeneration, you will see a thread-like structure, wherein you see this area is now started to disintegrate. Okay. So what we do is we cut the animal, we check for an earlier time point, which is the earliest point at which we can observe for regeneration, and then a later time point to see whether there is any improvement in the ability of the neuron to grow. Now, when there is mitochondria, you'll see growth cone being formed. And at a later time point, you'll see that the growth cone has elongated and you see a thread is being ejected in all the directions. and you see protrusions in all the directions. So this is like the nature of the growth cone to explore its surrounding and then move in the right direction. Okay. Now, when there are no mitochondria, in the earliest time point, you do not see any growth happening in these neurons. But at a later time point, you'll see some amount of growth cone formation in these. So this result sort of suggested to us that mitochondria, if present in the neuron, helps the neuron to start off this process of healing itself faster after its injury. And if mitochondria are removed or in a way are affected in the animal in one or the other form, it's functioning or if they are completely absent, the neurons are incapable or there is a very delayed start in this process of regeneration. Okay. The next question that we wanted to ask of how mitochondria is important and that I wanted to show everyone over here is the functionality of the neuron. Now, there must be some reason why mitochondria are present throughout the neuron, right? Is there a question?
Utilization has not been observed. Is the influx of mitochondria expanding?
So we don't really know that because the central nervous system, it has a lot of complex factors that act as inhibitory cues as well. So the question is that whether in the central nervous system, the inability of the neurons to regenerate, is it because of the loss of mitochondria? And the answer to that is yes as well as no, because the thing is in the central nervous system, there are a lot of inhibitory cues that prevent that scientists have found, which prevent the neurons from regenerating. At the same time, in certain cases, if there is any defect in the human itself, in the patient itself, in which the mitochondrial functioning because of some or the other problem or disease is lost, then they inherently will have decreased ability to regenerate the neurons. But a straight answer to this thing is that there are a lot of factors which inhibit the regeneration of the neurons in the central nervous system. And that is what precisely people are trying to find out. How would we enhance, or whenever a patient is subjected to injury, how would we enhance this, remove this inhibition and enhance these neurons to regenerate? Okay. So a major, yes.
Question, you mentioned 6 hours, 9 hours, so is it assumed that the growth cones will take just a few days to latch on and do the connection, or would it be a long, extensive process?
The process is quite long and extensive, but yeah, the question that's, I'm really sorry, I'm forgetting to repeat the question. So the question that sir has asked over here is that whether this process of forming the growth cone and then reforming the connections with the neighboring neuron, whether I'm sorry, can you repeat that?
Like
Is it
hours, days, or are you talking about a long?
Yeah. Does it happen within a matter of hours or in days, or is it a long and extensive process? So a short answer to this question is that it depends on the system. So the system that we are using here is C. elegans. They grow within a matter of hours, and their lifespan is like they live only for 23 days. And they turn from a kid to an adult in a matter of 2 days, 2.5 days to 3 days. So because of the shorter lifespan, whatever processes that we see also happen at a very faster pace. But in humans, this process would take weeks and months. That's okay. So the next and a very important question is to understand what impact does mitochondria has on the function of the neuron itself. Because see, the main function of the neuron is to take the information from its neighboring neuron or the stimulus that is provided through the external environment and then pass it to the brain. So do these mitochondria also contribute to this particular function? So we try to ask this question again using C. elegans, with the advantage that we have 2 neurons in this animal. So this is an image of an animal in which we put fluorescent proteins such that only these kind of neurons are highlighted under the microscope. These neurons are called as mechanosensory neurons. Now I'll break it down. what these neurons do is that they sense any mechanical stimulus that is provided to the body of the animal. So for example, when I touch a person, okay, you immediately come to know that your neighboring person has touched you. That is because of a set of sensory neurons that are present right below your skin, which relays this information to the head. In a similar manner, these animals also have such sensory neurons present in the body just below their skin. And these are the neurons which I'm talking about. So we have the animal, and to provide this mechanical stimulus, we use this apparatus that we call, which is a stick attached to an eyelash. Now, because the animal is quite small, right? You can't touch it with your finger. You need an eyelash to gently touch it. So that's why we use a very thin thing, that is the eyelash that we can get from our eye. We stick it to the stick, and then we keep harassing the worm front and back, front and back, front and back until it is annoyed with us. So what we do is we touch the animal, it gets irritated, it goes back. We touch the animal at the tail, it gets irritated, it comes ahead. So what happens here is that when you touch the animal at the head, I'll go back to this image, this neuron gets activated and it passes the information to its neurons downstream that, okay, there is a danger, you shouldn't be over here, go back. That's why the response is in the opposite direction. In a similar manner, when you touch the tail of the animal, the neuron that's present over here, this neuron gets activated, and that passes the same information to the neurons downstream, and now the animal moves in the opposite direction. So it's like a mechanism for the animal to protect itself from danger. So do you guys want to see in video how this happens?
Yes, yes.
Nice. So this is the eyelash, and this is how the animal is. Moves back and forth and back and forth. So this is a very simple way for us to understand that if the mitochondria or whatever changes that we do in the neuron, if they're important for the function of the neuron, then the ability of the animal to sense this touch would be affected, right? And this also helps us to extract the information of whether these processes are quite important for the neuron function itself. Okay. So again, the two systems that we use with mitochondria in the neuron and mitochondria absent from the neuron. And when we have the response, this is what you see, that the animal is responding and moving back and forth and back and forth. And when we don't have a response, this is what you see. It just does not bother. It will just go on doing its daily work. It does not bother whether you're touching it at the head or the tail. It will be like, it enters a nirvana mode. So what we do is that we make these changes and we calculate the responses that we get, or we extract the responses that we get from these animals under the changes that we make. Now what we see is that when there is mitochondria, all the animals give a very high response to the touch stimulus that we provide. But when there is no mitochondria, this response decreases very significantly, which suggests to us that the presence of mitochondria in the neurons is quite important for the neuron to function further. And currently there is a very high work that we in our lab, as well as other labs all over the world, are doing to understand or dissect how exactly a mitochondria contributes for the functioning of the neuron when it is present, say, for in the axon, as opposed to the cell body, as opposed to different parts of the neuron. So with this today, I briefly discussed with everyone that the functions of mitochondria and its importance or its contribution for contexts like regeneration and neuron communication. But apart from these, mitochondrial importance is also being explored using neurons as a system in the process of growth of the neuron. So neurons, when they start as a cell, they actually start from the cell body, and then there are projections emitted all over the. And then one of the projection becomes an axon, which is like the projection which passes information to the neighboring cell. So how does that happen? And a lot of study in the field have shown that if mitochondria are not present, then this ability of forming an axon is hindered very badly. And one more aspect, which is of physiological relevance for humans very greatly, is in the process of neurodegeneration. So I'm sure everyone would be aware of all the neurological diseases like Alzheimer's, wherein the neurons, brain cells are affected. Mitochondria is very extensively being studied in this entire field, wherein people have seen a very high correlation between these diseases, mouse models having these diseases, and the absence of mitochondria. But whether mitochondria actually is one of the factors that contribute to these diseases or no is something that is being explored in the field, so that we can use this as a therapeutic treatment to at least improve the neurological disorders that we are facing. So with this, I'm happy to take a lot of other questions. Anything.
Okay, we will take a few questions from the online audience. For you, we can have Jayan come back and ask questions. But for the online audience, before we shut the online session, let me tell you, and of course for you as well, that if you're listening to us online, the next session, which is the mid-September, September 15th, is Engineers Day in India. So our next session at Ruparel College will be looking at building detectors for large experiments in physics. That's coming up next as our Engineers Day special. And a month later over here, most likely we are going to be having someone who's joined TIFR recently talk about how chemotherapy actually helps in cancer treatment and some of the research in new molecules for chemotherapy. So that's coming up a month from now over here. That's the look ahead. Let me quickly take two questions which were here. Yeah, online audience, this is your chance to ask questions. So Abhinav had another question about, is the mitochondrial genome still functional?
Yes, it is. So whatever proteins that you see in the mitochondria, they are a mixture of two kinds of proteins. One which are imported from the cell itself, and ones which are produced by the mitochondria also. So that's why it has its own protein production unit. So yes, mitochondrial genome is still functional.
Okay, there's another question. Are mitochondria, do they actually, like when you poke the animal, does a mitochondria respond to the stress, or it's the mitochondria is not the sensor?
We are trying to understand that.
Okay. And Tony has a question on, is there any way to increase the amount of mitochondria in the body?
It was the same question, yeah. So when a short answer is yes and no, actually the answer is yes and no. So eat good and you will be healthy, and that's why you will get good healthy mitochondria. But actually the number of mitochondria depends on what kind of cells they are in, and if the cell has a high energy demand, there is an automatic amp up of mitochondrial production in these.
Okay, so on that note, let us, we will end our live streaming. But audience, please here in Prithvi, please join us for a cup of chai and come back and we can discuss further. Okay, we'll take all your questions. No worries. She's not running away. Oh, there is a question online. Okay, go ahead.
Okay.
Yeah, why does mitochondrial DNA come from the mother only? Exactly.
So I'll have to go back to the process of fertilization of egg and the sperm. So when the cell is zygote is formed, what happens is that the egg that comes from the mother has all of the cytoplasmic or all of the material that is from the mother, which includes mitochondria and all the proteins that are present in the cytoplasm of the egg. And when the sperm fertilizes the egg, all it does is that the DNA that comes from the father is injected in the cell. So that is why all of the cytoplasmic material is obtained from mother, and mitochondria being present in the cytoplasm of the oocyte is, I mean, the simplest answer is from the mother. That's
Okay.
Process doesn't allow mitochondria to come in.
sorry.
The process doesn't allow others mitochondria to come in.
Yeah.
What is mitochondrial dysfunction?
So mitochondrial dysfunction is just the inability of mitochondria to function. So the dysfunction is it can't function properly, so it can't produce ATP, it can't take in calcium, and whatever functions that I discussed.
Okay, on that note, I think we thank Sneha and stop the live stream.
Thank you
And
so
please
much.
join us for a cup of tea.